3 years ago. May 4. My time sense has changed. I hope
So far, so good?
Yeah, so far, so good.
Hello, welcome this evening. I'm Ryan Phelan, and I'm here with the pleasure of hopefully really bringing Andrew Hessel's book, The Genesis Machine, to life. We're not going to go through the whole book, but I wanted to say a little bit about Andrew and his background to kick this off. Andrew is a microbiologist and a geneticist. He is a Canadian transplant, I gather.
I am.
And he's an amazing entrepreneur. He's worked in 2 different companies, one in a biopharmaceutical company whose name you probably recognize called Amgen, and then later with Autodesk, which I assume was started here in the Bay Area, right?
It was.
In Sausalito. And he brings, I think, both a commercial and very much a nonprofit thinking through how this technology is going to help benefit humanity, which is not always what we see in commercial companies in the biopharmaceutical industry. And I think you've come to loggerheads with that more than once. Anyway, it's really a pleasure to be here. My background actually is been in personal health and what used to be referred to as personalized medicine and now sometimes precision medicine. And then I went from that area into wildlife conservation, bridging biotech and conservation. And my nonprofit organization is called Revive and Restore. So over to you, Andrew. How about a brief intro of what got you into this book and this whole topic?
I live synthetic biology. It's been my passion for over 20 years now, probably closer to 25. But I'm a first-time author, and I was really fortunate during the pandemic, while we were all taking a long extended break and staying at home, to get a phone call from my speaking agent, who was not very busy at that time, introducing me to another person in their community, Amy Webb. And I had never come across Amy before. And so immediately started reading up on her and checking on her. She's amazing. She has an institute called the Future Today Institute. She's a best-selling author, written some amazing books, TED Talks, etc. And we got on the phone together, and she said, "Andrew, I've got this book deal where we've been looking at gene editing technology. And what I've seen is it's really much bigger than that. Today, the field of synthetic biology is gene editing is part of it, but synthesizing genes and synthesizing organisms is becoming a big part of it." And after that short conversation, she said, "I'd like to write the book with you." And so for me, it was great, because never having written a book before, it was like having a famous director just say, "Would you like to make a movie with me?" I said yes immediately, and it's been a great, it was an easy and fun process writing the book, which a lot of authors tell me is not usually the case.
Well, I fear that authors talking about the process of writing their book is a little bit like people talking about their dreams. It's really interesting to the people who have been living and breathing it. But I think the meat of what you're talking about is something that is going to be incredibly interesting to everybody, because this is affecting all of us right now. And this being the development of biological code that can be edited and adapted for all kinds of implications, from human health to food and scarcity of resources that can be replenished, to wildlife conservation. So let's bring it home, I think, to a good example of how synthetic biology has shaped medicine. So can you give a couple of good examples?
My life has been a part of this. So I worked for a company that one of the first, the medicines it made were created by reprogramming cells to make the medicine. And the programs they wrote, and then they started doing this a long time ago, we could only write a little program, a very small program. It's almost like learning to write as a child. You don't sit down and write a novel as a child. You start with sentences. Back when the company I joined was really just learning how to write the first sentences using nucleic acids. And these sentences are important because they're like scripts. They tell the cell what to do. And in this case, they wrote a program that told the cell, make growth hormones. And these are hormones used by your body to literally direct your growth. You've probably heard of human growth hormone. That tells your body literally to get bigger. The growth hormones this company made told the bone marrow, make red blood cells. Really important. They wrote another program that's told the bone marrow, make white blood cells. And so I was really fortunate to join this company early days and see this incredible technology be born and utilized in a way that ultimately helped millions of people.
And was that Amgen?
It was Amgen. Yeah, it's still one of the first generation biotechs and an absolutely amazing company.
And so 25 years ago, what do you think?
Yeah, it was coming up to 30 years. It was 1995 when I joined them. And I came from a world that Amgen is an American company. I was working in Toronto, Canada. And it was a completely different world. Amgen taught me not only about the biopharmaceutical industry, which I just didn't know about. It also taught me about how aggressive American companies are relative to Canada. Like, to give you some context, Amgen spent more annually on research and development than all of Canada. And so I felt it was a dream job, and to work with this company. It was the best education I could have had in biopharma.
I bet. I think a classic example that is often used to help people understand the role of synthetic biology is the production of insulin. And maybe you can tell us that story.
Yeah, insulin. Any diabetics in the room?
Well,
you
no,
don't feel like, you can't do that.
But probably everybody here knows somebody.
Let's just say I should be diabetic because my favorite foods for many years were basically gummy worms and gummy bears and just anything gummy. So I should be a diabetic. I literally ate candy all the way here from my home in the Russian River. But insulin was used to be recovered from pancreases that were harvested from literally livestock, and we would have to. It was really this massive job of collecting organs and processing them just to get a little bit of this incredible hormone that saved lives.
And actually, I remember that they were. Pardon? Oh, sorry, I thought there was a question there. No. Anyway, it went from originally, I think it was with pigs that they were harvesting this. And then the big breakthrough was, well, they could use other domestic animals. And then finally, these scientists were saying, well, how else can we develop this instead of harvesting from wild or domestic animals?
And it really set off it really seeded the development of essentially genetic technologies to be able to reprogram cells to be able to produce insulin. It was one of the real success stories. And it was an incredible amount of work doing this type of programming at the time, because the technology that was used was called gene splicing. And it was very similar to what we did with film or even with magnetic tape at the time, where you had to start with physical media, cut it, and then splice it together to create a new story. It's very much like writing a ransom note. If you've ever, hopefully no one's done that in a professional capacity, but I actually tried to do this just to really understand how much work it is. And you can write anything with a ransom note if you have a stack of magazines, scissors, and glue, but it's really time consuming. Now try doing that with the nucleotides of DNA in a molecule that's just a couple nanometers wide. And just writing even a short program was an incredible scientific accomplishment, where many different, and you have to validate the work that you're doing, the edits that you're doing with a whole series of experiments, because you can't see any of this directly. So it really, really tricky work. I was really fortunate that I came into biotechnology just as we were starting to make the transition away from this gene splicing technology to gene synthesis technology, where to first of all write the code that you wanted to do, you just used essentially a word processor. You could really start to write code, and it was so simple back then because there just wasn't a lot of code, that you could literally use Microsoft Word or Notepad to write out the digital language of DNA, which is written in A's, T's, G's, and C's, not zeros and ones like computers. But there's only 4 letters to work with, and once you wrote your code, the challenge then was just how do you print and assemble that code, compile the code if you're into computers. How do you compile it from digital DNA into the molecular DNA that all biology runs with? And that's been, I was really, that this field has been percolating now for just about 30 years.
So you make a great analogy in your book about how the advances of the whole digital economy and the digital world has happened so rapidly in the lifetime of everybody in this room, from decades ago, from massive parallel processing computers to the mini computer to personalized computers, and now to everything in our iPhones and laptops. That analogy only goes so far when it comes to synthetic biology, or biocode, as you refer to it. And I'd like to, part of your book, you talk about the value network. And I would love for you to describe a little bit about the value network that we're all living in the digital age, and how that could be translated to biocode.
Well, let me ask a question that isn't medical. How many of you use a computer today for your work? Yeah, okay. For those of you online, and I know you're using a computer, we're looking at 95, almost 100%. Like we are now cyborgs. The value network of computers, which really started personally in the late 1970s, beginning 1980s, has changed society. It's changed our economies. It's changed just about every business practice. And it really hasn't taken that long. It's taken, less than 40 years for that transition to happen. And that was I lived that. I was one of the first people to start to buy these systems and use these systems every single day. And we were just kids. We were playing around. We weren't trying to build a business or anything. We were just learning and playing and sharing. And even though and I remember I woke up one day and I thought, well, do I want to work with these computers every day? But the change, the pace of change was so dramatic in computers at that time. Like the languages were popping up every day. There were completely new companies coming into the game every day. I couldn't afford to keep up with the pace of change.
But I think that's happening now.
It's still happening. It doesn't
you
stop.
feel like it's slowed down, though.
No. It's only accelerated.
Okay.
It's a Red Queen problem. It only accelerates and becomes more sophisticated. And we don't even think about the low-level stuff in computing very often anymore. But it's just layers and layers and layers of economy and networks and capability.
So that's the value network.
That is.
In the digital world. But in the biological world, I had a feeling in your book that you were frustrated that it wasn't advancing as fast.
It is. Biology is one of, well, first of all, we're all biological machines. We're all biological creatures. This is a living world. Biology is the technology that connects all of us and works so well that we don't pay any attention to it unless it's breaking. And so, but I've always looked, and I think it's because I was in love with computers in my early days. I always looked at biology. I started seeing the parallels. Wow, a computer is really much a lot like a cell, and software really is like a genome. And then I started to work with digital DNA as we started to sequence the first organisms, and we suddenly started to get files on computers of genetic code that needed to be analyzed and processed. I was right there at this transition and watched it emerge. And was fortunate enough to be working with one of the companies that really learned how to make a thriving business out of it. Amgen was Google in biotech, essentially. And but I am frustrated that it does take a while to build the foundations for more players to come in, more people to come in, more creativity to come in, and to start turning that into products that we all use every single day, like we do our computers, like we do our bodies. And so we're, just to, we were talking just in the green room. Synthetic biology is still under the radar. Like if you're an investor, if you're a creator, if you're just young and are looking for something incredibly interesting to do with your life, I really couldn't point you to anything more important and more powerful than synthetic biology, because we have to stand on the digital architecture. We have to stand on just biology, that has created the world around us. To get started, you have to understand some of the systems. Thankfully, they're pretty easy to understand because all life is built on the same architecture. It's not like you have to learn different paradigms for different organisms. They really are all built on the same architecture. Today we have this digital architecture that's now giving us insights and data is often in real time now at single cell levels about this architecture. That's connecting this new nervous system and sensory web. And now we're standing on that, and we're learning how to write code and program this incredible architecture to meet the needs of humanity.
So, Andrew, let's talk about some of the cool ways that it's affecting all of us right now. And so I'm going to ask a question. How many people here have tried cellular agriculture, whether it's Finless Foods or Impossible Burger? Can I see a show of hands? It's like almost everybody here. And that's just the start of what's happening in synthetic biology for changing food. And the implications are way beyond, I think, what many people have imagined of just what's going to change in our diet here, but how it's going to change things globally. You want to address that?
Well, and we've been eating genetically modified foods for a long time. If you've ever had a bag of Doritos, like most of the corn and soybeans, and that's synthetic biology with the old tools, with the cut and splice tools, the recombinant tools. Moving forward, the idea of doing this engineering now, you don't necessarily have to be a large ag biotech to start to do this work. If you decide that you want to go into your garden and start modifying, the tools are becoming accessible enough to do this at an individual level. Certainly at a level where you build a small team and get it funded because there's something creative you want to go do. But there's an inherent it is also one of the hardest fields to get going in, because biology, the synthetic biology makes the genetic engineering part of it easier. Now we have design tools and editors. We have DNA printers rather than having to do the cut and splice techniques. But there's still a lot of hands-on physical work with the biology, loading code into a cell, checking whether it's doing what you want. And of course, biology moves relatively slow compared to computers, where you can compile the code and get your results literally almost instantaneously. You make a change to an organism, you have to grow that organism. You have to be able to test it.
So probably even the bigger issue with all this is the social license to be doing this, and the lack of regulatory mechanisms for this. Do you want to address
Well,
that?
and we mentioned this in the book. What we're starting to see is this great acceleration. And in the early days of computing, there were no regulations about the internet or computers or networks. It was really the Wild West. And it was only when we started, in retrospect, we started to build the educational systems and the regulatory networks. We've always had the legal system. And if someone's doing something bad to you, we've always had, the ability to go and rein them in with the legal system in various ways. But when with biotech, it's just hard to get started. And it's going to take a little bit of it's going to take changing the framework of the regulatory systems we have in place today, with things like the USDA and the FDA and even the FBI, to really start to open this field up to more creativity, to more play, to more
Innovation.
yeah,
Just
innovation.
pure innovation.
Yeah.
But I don't want to make it sound too hard, because there's so much going on in this field. And I think that the assumption is that the regulatory mechanisms will catch up. And certainly this has happened in medicine. I mean, probably all of us have had the COVID vaccine, which we couldn't have had without it being fast tracked. And so I think there are plenty of exceptions that are going on where medicine is moving so fast, and it finds a way when it's needed.
Yeah, that was a big win, because we really greased a lot of the wheels in the regulatory architectures to push that through as quickly as we could. And I look at the mRNA vaccines that were produced during COVID as being the biggest advancement in vaccine technology that I've seen in my lifetime. And I'm even more excited about where it goes, because we describe some of this in detail in the book, but it really only took a couple of months to make the vaccine, and to make clinically pure vaccine that could go into trials.
But explain why, since you have a background in vaccine and working in cancer tumors. And
it was really quite many of you probably already know the story, but the very first report of this new of this new disease popped up on New Year's Eve in 2019. And so no one saw the news. Obviously, we were all too busy partying. But at the beginning of January 10th, the sequence of this new virus was published and was put into the virological community
by the Chinese.
By
Originally,
the Chinese, yeah, working with an Australian colleague to get the information out. Which was great. But what's interesting is already we've recognized that it's possible to make vaccines very quickly with synthetic biology. So there were groups that had already received grants, had been working with multiple agencies to be able to fast track vaccines. They were immediately like, oh, this is interesting. And it was a coronavirus, and other coronaviruses, SARS and MERS, had already caused outbreaks around the world that were significant. So they were primed to look for a new coronavirus, and they took the chance to start developing a vaccine just as a creative effort. Did that in a very short period of time, and then made the really gutsy move to put it into clinical development, into GMP manufacturing, essentially clinical grade manufacturing, just 10 days after that. So that's why we had vaccine that could go into trials in February of 2020. So this was really remarkable, because there was no real profit motive out, and we didn't even recognize if this was going to be a bad outbreak or pandemic at that time. So it was really just the pump was primed, the timing was there. It was the cost of making a vaccine had dropped significantly because of synthetic biology, that it really jump-started the whole process. And again, as time showed, we greased all the wheels in the regulatory machine, really rushed through the clinical trials as quickly as we could, not without compromising safety, and of course got our shots in the arm. But now that technology, now that we have pushed that through, we're so much better prepared for the next infectious agent, which is amazing. And in fact, DARPA has a program called Nucleic Acids on Demand worldwide, where they actually want to be able to make a vaccine in the field in something like 72 hours now. And so which is incredible. But the mRNA vaccine is really just a little bit of nucleic acid wrapped in a little bit of fat, a lipid, that is not particularly targeted to any cell. So it's just a quick injection, go. Your cell becomes the manufacturing plant. You don't need a big pharmaceutical company.
So, Andrew, you started a company called Humane Genomics that was focused on cancer and cancer vaccines. Can you tell us a little bit about that?
Yeah, this was a long process. When I left biopharma, I realized, wow, this is really an incredible technology, but making a drug takes a long time, and it's really expensive. And I needed a change. And so I left the company and I took a year in Thailand. And when I was in Thailand, I learned that it doesn't cost $1 billion to make a drug. People were making drugs all over the place, and it was pretty cheap. What I learned was, and I knew about this technology of synthetic biology because I'd been working in biopharma, and I realized I could see this future where it would be possible to design and build medicines really inexpensively, and maybe not take 10 years and $1 billion to get it through the regulatory process. And so the wellspring of money in genomics is cancer, because at the root of every cancer is a corrupted genome. Essentially, it corrupts all the cellular processes, and ultimately we get cancer. So I got this idea. Well, maybe I can make a company that's very open and collaborative, that uses synthetic biology to make a medicine that could be used to treat cancer, and we could do it very collaboratively. And then I saw this wonderful paper about something called synthetic oncolytic viruses, or oncolytic viruses. And it was really just viruses that were too weak to infect normal cells, but cancer cells are broken, so their defenses are broken. And these viruses could slip past the cancer cell's defenses and preferentially kill the cancer. And I thought, wow, that's fantastic. And maybe we could use synthetic biology to make those viruses. It turns out in 2002, the very first genome was written from scratch, and it was a virus genome. At the time, it wasn't a big virus. I think it was like 7,000 bits of code, nucleic acid bits. But it took about $500,000 and 18 months to do that genome synthesis and assembly. But that to me was like, wow, if we can do that, knowing that this is going to get faster, better, cheaper, like everything digital does, I realized making a virus was going to get pretty cheap. And making a virus that could hunt down and kill cancer cells specifically for individuals would be really amazing. It would be a groundbreaking change. It took over a decade to realize that vision. It took me about another 8 or 9 years to be able to make my first synthetic virus. And then we started working on synthetic cancer-fighting viruses and incorporated that company, I think it was 2018. And now it's operating in New York City. I'm just an advisor to it. It's moving along at a pretty incredible pace.
So help me out. In your book, you have a section about risk, like a lot on risk. Even though this guy is a complete optimist and bullish about the technology, you spent at least 50% talking about the downside. And one of the things that you mentioned in there is about trying to put a ban on gain of function research with viruses. And that seems like a disconnect with what you just said.
Well, there was a group of scientists that started doing what's called gain of function with viruses. I want to be you have to get really specific on some of these terms. Gain of function is if you make a cell do anything new of benefit, produce a protein, produce a medicine, that's a gain of function. You're changing the function. You can also get, less function. We don't talk about that as often. But when it comes to gain of function with a virus, you're essentially making changes that will make that virus either cause a worse disease or be more infectious, spread easier. Um, and there were whole lines of research where people were doing gain of function with viruses to try and understand what are the key parts of a virus and what changes need to happen for them to become more infectious or more deadly, so that they could get ahead of the next disease, the next pandemic. Today, I think given the pace of that type of work in the past, that type of research might have made sense. With synthetic biology, I think it makes less sense. And today, my biggest fear of these technologies is that it is, and I know from experience, is that it's relatively easy to design and build a virus from scratch because they have very small genomes. And you don't really need a manufacturing plant to produce a virus. You just need essentially a seed inoculum. And you can start to make virus particles. Viruses go through the populations of hosts almost in a biological chain reaction. And it's not one-to-one. A virus infecting a cell, let's just look at it as a bacterium, could produce hundreds of new virus particles in that bacterium. So it can cause this multiplicity of infection
Maybe we
that can
shouldn't talk
cause
about
it to
it in this
spread.
crowded room.
Do you remember the R naught that everyone talked about
Yeah.
in 2020? That's essentially
Yeah.
the how many people one person can infect. And if one person can infect one person, it doesn't really go anywhere. If you infect two, it kind of breaks even. Anything above that is just it's just an accelerating exponential.
Okay, well, let's talk about the upside. So
Yes.
one of the ones that you get very passionate about, I should say, is around sex and reproductive opportunities, given the new biology. Do you want to give people a little sense of where that's going?
Yeah, in fact, again, I live this. The book tells a few anecdotes. I was not going to have children, and I was a pretty tenacious teenager, and went to my doctor when I was 18 and said, "Fix me," because I really did not want to have kids. And it's just I was kind of like Benjamin Button. I was kind of old from a really young age, and looked at my parents and just realized these people probably shouldn't have had children without a license. So I really didn't want to have kids. Let's just say that life has a way of changing. And by the time I was almost 50 when I met my now wife.
I have a feeling this goes back to Thailand.
No.
Oh, okay.
No,
okay,
it doesn't.
Okay. I heard about that here in Thailand.
No, it goes back to a TED conference, actually. But my now wife literally picked a biological lock in my head, and I realized I want to have kids. By the time you've had your taps turned off for that long, let's just say that things don't work that well. So this is a little intimate. The short version is our kids were produced in labs with IVF. It's not uncommon. Millions and millions of kids are produced in IVF. But it was really our understanding of cell biology developed perfected in the 1970s that opened the door for people to have kids if they were having problems. And today we understand the human reproductive process so well that there's multiple interventions that we can do. So IVF is a machine now, and a lightly regulated one at that. But now we're starting to get to the point where these technologies can take us to the next level. For example, IVF, it's really hard to get eggs. Really hard to get eggs. It is. My wife worked hard to get the handful of eggs that we needed to produce our kids. There is a technology on deck called in vitro gametogenesis, which is a fancy way of saying making eggs and sperm in the lab. And that's going to be a massive game changer for IVF. When IVG starts working, IVF could be $1,000 and something you could do in an afternoon.
Well, so not only that, talk about sex selection, because I think any gay couple might be particularly interested in that.
Well, we're already getting to the point where we have 3 parent babies because some people have mitochondrial diseases. The mitochondria from the sperm never go into the egg. So it's a maternally derived organelle in the cell. That's pretty amazing. So we're doing that. We can already start to manipulate the genomes well enough that you could have same-sex parents producing an embryo. That has not been done for humans yet. You can have 0 parent babies, clones, which are just identical twins separated in time.
Yeah, but you can't do that.
Not yet. Hasn't been done yet. But you can argue that there's less ethical argument there than there would be with making a CRISPR edited baby, and that's already happened. It certainly wasn't something that was widely accepted or approved. In fact, the researcher that did that work in China just got out of jail, I believe, 2 weeks ago.
We never hear about those 2 children. Are they healthy?
We just don't hear about those children. So they've never been actually verified as their edits have not been verified by the West.
Their edits were to make them immune to HIV, as I recall.
Yes. Except that and it's funny. I've actually spoken about this a few times because I really just believe I really like the power of gene editing technology. For example, if it's genetic surgery. So if an embryo, if that embryo has a serious error that would cause a miscarriage or a really horrible genetic disease, this technology could effectively do the surgery to repair that pre-implantation.
Right, but this is not future. This is current.
This is current,
Yeah.
and the technologies are current. The thing that was the reason why there was kind of this reflexive response to this was actually an enhancement of the human genome. It was adding a function, a resistance to a virus, and potential that was not essential for life. So, so I really just look at the researcher as being badly advised, because if he had used CRISPR technology to do genetic surgery on an embryo that allowed that embryo to come to term, not miscarry, like there was a serious error, then even if he had cut some corners and kind of pushed the limits, I think there would have been a moral acceptance that what he did was really appropriate. And the parents would be so happy because they would have their child.
Yeah, so this again is the Chinese geneticist that edited the HIV immunization. And I think that part of that challenge was that he didn't get informed consent. So there was no informed consent with the multiple couples that went through it. So it wasn't just, it was kind of a spurious gene edit. It
Right.
was the whole process.
And again, without having full transparency, it's really hard to know all the sides in the story.
Right.
It is interesting. Well, you have to rewind back to the 1970s with the first IVF baby too. Louise Brown was the first IVF baby born in 1978. The parents didn't have informed consent either. Like, the mother was a little surprised when suddenly
I had no idea.
they had no idea that they were now going to have, be on the front page of every newspaper in the world. But the thing is, kids are just kids. And even though, we were taking the reproductive process and putting it in the lab, people realized it's not that sensational. We're really just mastering the biology. What's interesting today is we're going beyond what nature could do biologically. We could, we can now do things like enhancements and surgeries. I talk about one day we may just synthesize chromosome 24. And just preload it with software that we may need in our life, because we still don't have the technology to upgrade our genomes in each individual cell in our body. And we have 50 trillion cells, and they're constantly turning over. So we just don't have the tools to do that type of.
But it's coming.
Well, not necessarily. We even in the lab, trying to move a big chunk of DNA into the nucleus of a cell is a problem we haven't solved. So trying to do that in vivo for 50 trillion cells,
It might be a small chunk.
putting in small chunks is fine, but if you want to put in a whole new chromosome or a lot of code, right now
Or
the
a
only way
genetic
to do that
vaccine, a
is,
heritable
yeah.
vaccine you could do. Theoretically.
That's a small amount
Yeah.
of code though, so that could be delivered with a virus particle, for example. But we don't have a way to drop in a whole chromosome yet.
Right. Well, one of the ways that I've been dealing with this reproductive challenge is with the cloning of wildlife for conservation. And last year, we've been working with U.S. Fish and Wildlife Service on America's most endangered mammal, which is a black-footed ferret. And this, the history behind this particular species has been that it's been an ecosystem engineer across the American prairie, and but because of the way agriculture is developed in the American prairie and because of habitat change, they were driven to extinction. And by 1980, they were pretty much extinct in the wild. At least they were thought to be completely extinct in the wild, when one colony of 18 different ferrets were found, and they were brought in, and they've been in captive breeding with the U.S. Fish and Wildlife Service for now multiple decades. Well, it turns out there were only 7 founders. And so that means only 7 individuals of those 18 ever went on to breed, which now, 40 years later and multiple generations, all 300 ferrets in the wild and 300 in captivity are all basically siblings. That's not good. And conservationists call it the extinction vortex, when small populations of endangered species start to inbreed and they become increasingly susceptible to disease, and they start having reproductive problems, and then that's the spiral down into extinction. And this really does happen with all these endangered species, where the genetic variation just gets lost. Well, Fish and Wildlife Service heard about our organization trying to bring biotech to conservation, and they contacted me and they asked this question: well, what can biotech do for our black-footed ferrets that we're not trying? And it turns out, they told me there were frozen cell lines that had been banked 35 years ago from 2 individuals that never contributed to the gene pool. And that meant there could have been new founders. So Revive and Restore did the genomic sequencing, and we found incredibly unique genetic variation that had just been lost over time. And last year, we actually cloned one of the black-footed ferrets, and Elizabeth Ann, who's now exactly 1 year old, is now going to become the 8th founder when she starts to breed. And she will bring in 10 times more genetic diversity back into that population.
Do you see using synthetic biology in the future to introduce new variation into the genome?
Well, we do. And partly it's because many species are incredibly susceptible to invasive pathogens. So sylvatic plague is a good example that affects the black-footed ferret and many, many other species. And they have no inherent resilience to it. And again, as these populations get smaller, they become more vulnerable. And I think one of the things that's challenging for people is they don't really realize how much of nature we intervene in all the time. That's how a lot of wildlife exists on this planet. And so it starts to feel like you're tinkering with nature, but the truth is we're always meddling in nature, and we want to do it for good, not for evil. So how far do you go, and how do you make those changes? But one of them would be to engineer in resilience.
Yeah, I think that's really important because as we know, we're threatening so many different organisms today, just because we've been so successful, and because we really haven't been good stewards of the ecosystems. I think that awareness that that we have to understand. Well, we used to catalog nature pretty well. But now we realize we really have to help it. We really have to champion it. That there's incredible diversity that we're just going to lose if we don't. By the way, if you haven't looked at the frozen zoo, You really need to, because it is the coolest zoo that I
I
can
don't think you
imagine.
get to look at it.
No,
Yeah.
but just the fact that we have all these cells banked
Yeah.
from all these different organisms is remarkable.
Well, an exciting thing that came out of our research, and once Fish and Wildlife Service saw this incredibly charming, adorable little black-footed ferret that was born, a healthy clone, despite many of the myths about cloning that have been debunked, they asked me the question of, well, what else should we be doing in this area? And I said, we'll start biobanking, cryopreserving now the 1,500 endangered US species alone in the US. And when I did the research on it, less than 17% of all critically endangered US species have ever been cryopreserved. That means when they're gone, their genomes are gone, their living cellular tissue that could be banked is gone, and there's no way to help at that point.
See, we're already at the point today that we don't need even a living cell to recreate some cells, to boot up the very first
I'm not
cell.
so sure about that.
Well, the very first examples are bacterial, and we don't have a lot of examples yet because there's been exactly, I believe, 3 cells built from scratch, just 3. That's how early this field is. But it's still pretty remarkable that you can write a genome of a bacterium and boot it up, and by just putting it into a bacterium where its genome has essentially been erased. And now you can create that bacterium again. We're not building cells from scratch yet, but we're learning how to program them from scratch. And I like to believe, with faster, better, cheaper, and the fact that we're all working on a common architecture, that it will only be a small number of decades before nothing has to go extinct anymore. That would be really fantastic. And that we get a growing library of organisms that we can kind of print and boot on demand. Just in the life sciences, that would be really important. Because I used to work with bacteria. Now, you can't just look at a bacterium and know exactly what you have. In some cases, you can do it. You can recognize the colony. They have different morphologies. But really, to understand what you're working with, you need the sequence. You need to understand if it's mutated, if it's changed. To get a verified organism, you have to go to a biobank. And so my goal, like if I go to buy an E. coli specimen from a biobank, it's approximately $500 with shipping. So my goal, what I'd love to see, one of the waypoints in the future that I keep an eye out for, is when we can print an E. coli bacterium from scratch, from a computer file, for $500. When we can do that, we have full genetic control over the metabolism of that organism, and that's an organism we can all play with.
This is one playful guy.
I want my kids to be able to program bacteria the way kids today learn how to program an Arduino or program a computer. I really want that. They don't necessarily have to go and cure cancer tomorrow. They don't necessarily have to go and produce
Maybe in a few
product. But I want them to understand this technology, not be afraid of it, and to be able to play with it and be able to learn it, and to do useful things. Because I really look at synthetic biology as just programming cells.
Hey, let's open it up to questions.
Please.
Hello.
Hey guys.
I think they're going to pass around microphones, so we'll just wait.
Is there any way we can get the lights where we can see the audience?
Probably not.
Probably not.
Yeah.
Okay. I'm so sorry.
Okay. Hey, I'm Stephen Hubbard. I work in these industries and know what you guys are talking about. I also am very aware that the Bay Area is a bubble, and Bay Area biotech is an even smaller bubble. And so I'm curious about the experiences that both of you have had talking about this book or Revive and Restore, and about these general technologies to wider and wider audiences. In particular, I'm really curious what we in this field, or perhaps the people who've gotten to hear you talk about it, can do to help people feel more comfortable, right? When you say you want your kids to play with writing the genomes of organisms, I get that. I want that too. But I think there's a lot of people out there in the world who would be like, whoa, 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 crazy, weird, ick. All right. What are you guys doing when you try to help people become more comfortable with these ideas, and how's it going?
Well, I wrote a book, but more than that, I try and I do a lot of public speaking, but I try and champion the things that I really believe are going to be important. It's hard to do biology at home in some ways because labs require certain equipment to do test and measurement. It still requires a fair number of specialized chemicals and reagents that you just can't order off of Amazon. And nor should every home be a lab. So I've really been supporting the idea of biofoundries, which are kind of like cloud labs that you can program to run experiments. So they're controlled environments, they're sophisticated, they have really powerful tools and reagents, and robots to be able to do some of these experiments and get really high data streams back from that. And right now there's very few examples of these labs, but they're starting to appear. And there's some that are even accessible just to anyone in this room for doing simple experiments. And my favorite one is BioArtBot. BioArtBot.org is essentially in a lab in Oakland, but it is a robot that you can program to make pictures with living organisms that produce different color pigments. It's a really graceful entry point. But in general, the having secure facilities to program. We use cloud computing every day. I think cloud labs are going to be a big part of it. There's contract research organizations, pardon me, that I think will also be a part of it, where you spend more time designing the experiment than running the experiment. And in general, I think that we're going to start to see schools with facilities that are just accessible to a growing and diversity of students. Not everyone has to go into biosciences to start to work with these organisms. I think we'll see a lot of this coming together, like we've seen with the student program that has been based out of MIT for almost 20 years now. The International Genetically Engineered Machines program. That's still largely mentored using facilities, very professional grade facilities, but it has created a community now of students that I believe has surpassed 50,000 globally, and is just doing incredible work, opening up this field to a whole new generation.
I think, Stephen, I'd answer your question. In conservation, we're really trying to demonstrate successful cases, successful examples of incorporating these new technologies for wildlife conservation. There's nothing like seeing an adorable, successful, unique genetic variation in one of these threatened populations. It goes a long way. The American chestnut tree is another example, which for 30 years, I think probably most everyone knows about the demise of the American chestnut tree on the East Coast, yes? It used to be in the billions on the East Coast until the chestnut blight coming in from the botanical trade absolutely wiped them out. And these were the redwoods of the East Coast. I mean, fine musical instruments and furniture and livelihood from the chestnuts were, rained down on people there. For 30 years, the friends of the American chestnut tree have been trying to figure out how to backbreed in some kind of resilience into it, to no avail. I mean, they would try and they'd establish populations, and then 15 years later, these trees would succumb to the blight again. But now a team of researchers at State University New York, using one transgene from wheat, have been able to insert that into the genome and ensure that it is blight resistant. And it's an incredible story. It's got a dedicated community of people willing to work hard to preserve and are ready to transplant it as soon as they can meet federal regulatory guidelines to bring it forward. So I think when we start to see examples like that, it's the equivalent of genetically modified organisms that are now benefiting conservation that I think will be a real game changer for the public. Other questions?
Yeah.
Yes.
It's good to hear that the number of ferrets has gone up from 300 to, I'm here on your, at the back of the room.
Okay, thank you.
So it's good to hear that the number of ferrets has gone up from 300 to 370 now. It is also good to hear that the frozen zoo at San Diego Zoo now holds 800 species. But there are less than a dozen frozen zoos around the world. Why is that?
Well, this is the moral hazard argument that comes up when you use a technology and people think, if it's successful, does that mean we don't have to preserve nature anymore? Like you just said, we may not need living cells. We may not need the living organism if it's so easy to bring it back. I think that's a big issue. It's stymied many efforts in this way, and it's an absolutely vapid response to it because it's so important to preserve life. But funding for frozen zoos has never been significant, and I'm hoping to help change that around. But I think partly it's a denial of how fast we're losing species as well. And it's much easier to not really worry about it than to actually build something for the future.
Really, we should be putting these in every country because it's part of
their heritage.
Their heritage.
Absolutely.
Exactly.
And there are efforts. They're just, there's not very many, like you said. Somebody else over here had a hand.
Yeah, I see certainly benefits of biodiversity preservation. But when you're talking about an ease to create viruses and bacteria, then there is a kind of a natural concern to say, whenever you experiment, there's always failures. There's always unintended consequence that comes up. There are mutations that could come up. How do you handle, or how does the genetic community or biology community look at how do you deal with people who are either unintentionally creating bacterium or viruses that are detrimental to life, or deliberately creating bacterial and viruses that are so easy to now create that are detrimental to our world as we know it?
And you've hit the nail on the head. I think put this way, I gave presentations for almost a decade, really speaking about the inherent risk of synthetic biology and engineering viruses and bacteria, things that we know can harm us, and how the technology was moving faster than the regulation. Then COVID hit, and I thought, finally, we will pull together and recognize that this is an alien invader. It was the first pandemic in modern history. We'd pull together and start to figure out how to defend humanity against this organism. That didn't quite happen. So we're not there yet. This is one of the reasons why I think we really need to slow, if not immediately stop, a lot of virus engineering at this point, except for, and keep it as transparent as possible if we are doing this work, because we have that opportunity. This is a field that is still starting up. Remember, there's always churn going on and new bacteria, new viruses and mutations happening at a global scale today. In the synthetic biology world, it's a drop in the ocean. So we have the opportunity today to think about, how do we want to do this? How do we do the good things with this, like make the virus that attacks the cancer cell or delivers the gene therapy to the patient that needs it, and stop prevent as many accidents as possible, and certainly identify nefarious actors. And we don't have that framework yet.
Well, I want to, say a few things on that one, because I do understand that the FBI closely monitors all virus production. So people should not feel like it's a completely rogue environment. This is, these are registered, and this happens internationally as well. I mean, Homeland Security and everything has been on this for quite a while.
But it's a select group of viruses. It's and toxins. It's only a list of 50 or 60 organisms, and so there's a lot of room to slip through the cracks.
Yeah. So there's always room to improve safeguards, I'm sure. But I also feel like there's been some suggestion of banning this kind of research at all. And I think when you move too fast, you make mistakes. And I think my husband, Stuart, said this very well. He said, but when you move slow and steady, you learn. And that's what you want to be doing in all of this experimentation. You don't want to stop and start. You want to go slow and steady and iterate and learn and take all the safety methodical steps and create protocols along the way. If you can do all that, you can really advance the field. But moving too fast is never good, and moving in a stop and start is not.
Yeah, I think I'm basing it on Asilomar, which was in the 19, when we first started genetic engineering. We did pause for a minute to really figure out the right framework for moving forward. I think when it comes to viruses and engineered viruses in particular, I think we really need to slow it down and make a framework. I believe the framework that will allow us to move forward safely and well is a transparent framework. I believe that, but we haven't implemented that framework yet.
We have another question.
Well, it's somewhat related because I had a similar concern. But I think it's easy. The good stuff is easy. Everyone knows what the good stuff is. Everyone sees the good stuff and says, yeah, that's great. Saving black-footed otters, I mean, terrific. Cancer curing, awesome. People who are engineering viruses to kill people, they're bad. They need to be dealt with. And we have mechanisms for the bad, and we have mechanisms for the good. It's the gray area in the middle that's where I want to put you guys on the spot and ask you guys, okay, so what are the guidelines? What should we be doing? What are your recommendations as people in the industry to say, how do we protect people from their own, I want 6 foot tall boys with blue eyes, and this is what we're going to breed for. I mean, this is, people make bad decisions, and the tools seem to be getting easier and easier to use to make those bad decisions. Not that they're going to go kill a bunch of people and have some mass extinction virus, but that they're making bad, moral choices.
Choices.
How do you so what's the framework? You keep talking about a framework. What is it? What do you guys recommend?
Yeah, so I want to be clear that his book actually goes into the risks here and does different scenarios, and they're even more bullish than what you mentioned, more extravagant, because he's a very playful guy. But I think that in terms of with conservation, there are good mechanisms in place, and there's lots of regulatory steps to it, and there are plenty of protocols of how do you intervene in nature. In human medicine, there are also incredible regulatory environments that from clinical trials and everything else. So before anyone is going to be able to genome edit their children in any of these new trait selections and enhancements, in any scale, it will become part of human medicine and be regulated. I don't have a doubt about that. It doesn't stop people from making bad choices. And that, I think, is just going to be up to human nature. But you might want to answer this a different way.
Yeah, I'm not so much worried about the human part, because I agree with Ryan. I think it gets really interesting in the gray area of things that, we feel we have dominion over all life. I worry about intelligent cats. Yeah, well, that's just it. Like, then I mean, like IQ of 150. Like, if I have this one slide in my deck of a cat at a computer, and I tell people that, we've been making them smarter, and now they're, and a lot of people believe it. So, human intelligence and cat intelligence the inflection could be a little strange. But no, I see what you mean. I think we will be pioneering the processes as we move forward. We'll find out very quickly, because let's face it, we're all digital today. We get our information really fast. Things that are weird are front page news. So I think, the exceptional signals are going to pop up, and we'll start to talk about them and sort them out pretty quickly. Thankfully, we're just at the beginning of this field. Again, we've only made 3 microbial genomes and probably less than 100 virus genomes, and those were really just digital clones. We've only just now started building out the application space there. So it's very early. But we have this experience now that we created the web, and we see how weird it can get. And that's our human intentions and motivations and actions, really put on, put on display. So we shouldn't be naive. And part of the reason why we're happy with this book, Amy and I are happy with this book, is it's a conversation starter. It's punching through the immune system. It's often the first time people have even heard the term synthetic biology or thought about some of these scenarios. So it's really starting to get the conversations going and thinking about that.
One of the things I love about the work that you've done in this area, Andrew, is that certainly in my field in conservation, people always talk about what about the unintended consequences of these technologies. They never start to talk about the intended consequences. What are all the benefits that can happen to society dealing with climate change and species loss and everything else? And trying to flip that narrative, helping people think through a first reaction of how do we get what we really can intend to do with these new technologies and make them a benefit, I think is so critical. We have a lot of imagination for dark scenarios. It's very easy to imagine, and I think your book is full of them, of dark scenarios of bad actors. But I think that it's not that they don't exist, but the upside is a lot bigger than I think people think. I think there's another
Yes.
question.
I have a question about the roles and responsibilities in the ecosystem. So what do you think, what are the roles of the academia and the research institutes, and then on the other hand, the VC-backed startup companies, and where should the next breakthroughs in the biotech field, or where should they be coming from?
Oh, that's a great question. I don't have an answer for it. I know we're getting innovation from all fronts. On the academic side, there's almost a breakthrough a week happening now in cell molecular biology, genetics, etc. Just a recent issue of Science just published this incredibly detailed study of gene expression in basically every human cell from all of our tissues. And this is work that requires teams of 50, 100 or more people doing incredibly precise work. This is happening almost on a weekly basis if you're reading the scientific literature. And then here in the Bay Area, the number of biotech companies that have popped up and been funded just in the last couple of years is astounding. In 2021, SynBioBeta, now rebranded as Built with Biology, which looks at the industry hub, noted that something like $18 billion was raised for synthetic biology startups in 2021, which was more than the previous decade combined. So we're starting to see an acceleration. Innovation is coming from all corners, sometimes from very young innovators, just coming up with incredible ideas and putting them into play. I noticed that on an iGEM team was part of a recent paper that I thought was interesting. And just again, teams are becoming part of the whole ecosystem. So how all this plays out, I think remains to be seen. But it's going to take everyone participating. This is a technology like computing, like your own body, that will touch everyone and basically every industry over the coming decades.
I want to add to that one of the hardest hit areas of climate change is coral reefs all over the world. And when I started Revive and Restore 10 years ago, I would talk to coral biologists of what are we going to do about the bleaching of the corals? Is there a game plan here? Like, who's working in this area? And 9 out of 10 marine biologists I spoke with said it's so dire. All we can do is try to protect what we have. It's so dire. And now in the last 5 years, I've seen a game change where they're actually feeling empowered that there are new technologies from proteomics and from even gene editing potential, or certainly for gene selection for corals that will withstand warming waters that might fare better until we can actually lower the ocean temperature. And so I think that even tried and true conservationists that have been just protecting species are now starting to think about innovative solutions. And that's really becoming ubiquitous in conservation. How do we actually intervene and bring in innovation?
So I've got one for you. So I remember the first time I saw Jurassic Park, I started thinking about, okay, I'm ignorant enough to know, I'm so ignorant that I don't know if it's plausible, but it certainly sounded plausible the way they described it.
Yeah.
So I'm just curious, where did they get it right? Where was it wrong? And are you guys out buying up lots of land and building fences right now because it is actually possible?
And well, I'm going to take that one. I can start by saying you cannot clone from stone. So no dinosaurs are coming back. So
Even if there's amber that
no,
had a little DNA
they're not.
in it? Okay.
They're not. But you can. Pardon? I think we'll probably get a mammoth-like Asian elephant that is cold adapted back, and I do think they may go into Siberia at some point. But all bets are off right now with Russia. But seriously, there are scientists now with private capital going into this. We worked with George Church's lab for many years on it, and I helped identify some of the initial cold adaptive traits that would be needed. But Jurassic Park, I don't think so. It was a pretty good movie, not great, but it was fiction. There's one other thing in there, which is that people always say that, when humans intervene, they always get it wrong. And I think that film helped create this myth, this dystopic myth, that how seriously we screw things up. And I just don't believe that's true. I actually have done a lot of the research on how often humans intervene in nature and get it right. And it's pretty significant. Oh my gosh. I
Yeah.
think,
Right here.
Yeah.
Okay.
Hi.
Hello, how are you? I want to talk about houseplants, how people are connected to the variegated plant world, discoloration, and synthetic biology. Biology has a connection to it.
I think houseplants and houseplant engineering is going to be massive. There's already this one researcher in New York, Sebastian Cachoba. I'll probably butcher his last name, but he describes himself as a molecular florist. He works out of a bedroom in his apartment, and he does some of the most incredible work just engineering plants using these tools that are becoming more accessible, that even MIT has tapped into him as a resource. He's got a Patreon. He's incredible. So I would love to see more of that. I'd love to see more kids in gardens. As we've urbanized, we've kind of lost our connection to the land and livestock and to the stars. I would love to see more of just people playing with biology at home. My daughter came back from grade school the other day, and she had a radish that they've been growing, and it's like it's magic to them. But when you can start doing this with intention and putting in new functions, most people aren't afraid of plants taking over the world. which is good. It's probably one of those safer sandboxes to start to experiment with. But let's just say it's an area that I'd just love to see flourish.
I have a comment. Can you hear me?
Yes, please.
I think recently someone did actually transfer our entire chromosome. I think it was done in diatoms. But the question I think I want to ask is, why don't we have GM cannabis yet?
GM what?
Cannabis.
Oh, cannabis.
I feel like that's one thing I really want to see a lot of, wild varieties. And then why don't we have that yet?
Well, I can I take this one?
Oh, sure, go for it.
So, the problem, one of the problems that I see is the demand for cannabis type products can grow to global scale. And you just don't want to grow that many plants and process that many plants. A lot of this has been done in greenhouses rather than outdoors, just to really control the environment and the production. I have no experience. I've never grown a cannabis plant in my life, but I was really fascinated by the people that were doing the sequencing of the plants and starting to move the metabolic pathways for THC and the various cannabinoids into yeast. And this has been going on for decades now. There's multiple companies that do this. Now, if you want to get high grades cannabinoids and THC at a scale that you can supply the world because you've got a cannabis drink, you can do that now at pharmaceutical purity because of some of the work that's been going. In general, yeast, is produce the active ingredients in all the bottles here by the bar. All of those are just different flavors, only one active ingredient. But we're on the cusp now of making the first fully synthetic yeast, which means you have bit level control over the entire genome. And you can bet that there are going to be millions of different new applications coming out of yeast. I used to joke about having cancer fighting beer and intelligence increasing beer or wine. There are so many possibilities here moving forward, and yeast is the manufacturing chassis that is available to basically everyone at limited scale, because you can make breads, you can make beers and other fluids at home.
Anyway,
So-
with cannabinoids, it is coming, and it's
Yeah.
coming in part because of the need to actually verify the level
Of the active
of the
compound.
active ingredient,
Yeah.
which is, you can go and get anything, whether it's your cosmetic cream or, arthritis cream or whatever it is, and you have no idea that there's any effective product in it. So it is going to get regulated, and the way it will be was through synthetic biology.
Yeah, there's synthetic neurobiology is just starting to open up. It was an area of interest when we first completed the genome, because when we completed the human genome, we also basically got the program for the human brain. We don't know how to manipulate it yet, but again, all the clues are in the code. That's why I think working at the level of genomics is so fantastic. It's like reading the source code on a program.
Yes, Stephen.
So speaking of cannabinoids, the question is about snake oil. There's so much in this field where there are people with PhDs who are saying, we're going to make this new product. It's going to be altering your microbiome, or it's a cream with a new ingredient that's going to really be good for your skin. And how do you evaluate the efficacy of something that's coming out of a technology that no one really knows about so much? And my worry is that there's going to be 10 snake oil products for the one that really works, and it leads to mass consumer distrust of anything coming out of synthetic biology.
So just in case, because he wasn't microphoned, if you didn't hear the question online, he's asking about how do you stop the snake oil? And really look at efficacy. Well, we do have an FDA, but that's not. It doesn't really regulate. Does that cosmetic cream you're putting on your skin really work to do what stop wrinkles? Yeah. So one of the guidelines I use is if you can't see the change or feel the change, there might be no change. But we are getting a lot better at molecular assays. And again, what happens in a dish or what happens in a chromatograph is not necessarily going to correlate to real effects. But I think in general, we have whole lines of products that we take because without any demonstrated
Because of
efficacy.
wishful
Vitamins,
thinking.
like you walk in and vitamins, are basically just passed through your body. So just buyer beware.
But what I like about that question, I think synthetic biology might actually start to give insight to, which is that we could actually start to understand on a molecular level what you're getting. Are there really, the essential ingredients that you want in it? The beauty of genome editing is that you can be so much more precise instead of this random, breeding that we do with plants. You can actually engineer a single gene and get exactly what you want, and you have theoretically the backup record of what that genetic change is. So I think it'll actually be a good thing for the snake oils of the world. Put them out of business.
Well, and we're already starting to see even one of the groups that I had worked with in the last few years is a cosmetics company. They're starting to really look at how do we make functional cosmetics. And one of the groups that I really like is just in acne, where you do get to see the change very quickly using some of these ingredients. In fact, they're using a virus called a phage that attacks the bacterium that cause acne very precisely. Really fascinating stuff, but that's actually not engineering the virus. It's just growing and cultivating it, the one that's specific for the bacterium. But eventually we'll start to tune this more and more. I love the idea that again, we're going to have more and more people coming into life science, just as we've had more and more people come into computing, but we're all working on the same platform. The cellular platform has been conserved for 4 billion years, the basic machinery. So the knowledge set will grow very quickly because it could potentially advance at the pace of software. With computers, you had to go and design new chips, build new chip fabs to go and build the chips and figure out products to put those chips in. Synthetic biology, once it gets going, and we figure out how to paint the right lines on the road, could really accelerate quickly because it is really software driven.
Yeah, I would say that's present tense, though. You're making it sound way too future. I think we're here. Yes.
One question. What do you think of the future of
CRISPR? You go ahead.
CRISPR is a gene editing technology that is revolutionizing so many areas of life science right now. I think it's going to keep delivering both incredible breakthrough results in the labs. I think it's going to start moving into clinical use for a lot of different treatments. But I personally look at it as a transitional technology, because eventually it's still using a molecular system to make edits in large DNA. And eventually, I believe we'll be able to synthesize even large genomes with this technology, but that's decades away. So it's a transitional technology that is really powerful, and the use cases are still being explored every single day.
Yes.
The basic elements can be made right fairly easily, yeah. But it sounds like you're a biohacker, and if you're not, you might want to check that out as a profession. I think we've hit 8 o'clock, so we're kind of coming up on our time. I want to personally thank everyone for your attention and questions and interest in this. Thank you so much. And Ryan, thank you. It's a pleasure.
And
being with
Andrew,
you.
thank you for your book and for the conversation.
Thank you.